Glucose transport. Glucose is a polar molecule, but the cell membrane is a lipid bilayer, which is nonpolar. The polar substances will not cross the cell membrane, so we need some kind of carrier in the cell membrane for the transport of glucose. Glucose enters inside the cell by carrier-mediated transport in the form of active transport and facilitative diffusion by means of glucose transporters, also known as GLUT. Carrier-mediated is divided into active, which is against the concentration gradient and unidirectional, and facilitative diffusion, or GLUT, which is down the concentration gradient and bidirectional. Active or secondary transport. It is also known as sodium-dependent glucose transporters. SGLT is coupled with sodium-potassium ATPase pump and are of two types. Sodium-dependent glucose transporter 1 are present in small intestine and renal tubule. They help in absorption of glucose and galactose. Sodium-dependent glucose transporter 2 are present in renal tubules and help in absorption of glucose. Clinical correlation. Renal glycosuria. Isolated glycosuria in the presence of a normal blood glucose concentration due to mutations in SLC5A2 gene that encodes high-capacity sodium glucose cotransporter SGLT2 in the proximal renal tubule. Facilitative diffusion or sodium-independent glucose transporters, GLUTs. It acts by ping-pong mechanism. Ping-pong mechanism of glucose transporters exist in two principal conformations of carrier protein. Ping state, exposed to high concentrations of solute and bind it to specific sites on the carrier protein. Pong state, binding of carrier protein in ping state induces a conformational change that exposes the carrier to a lower concentration of solute. Glucose is transported down the concentration gradient. The following are the glucose transporters, their location, and their function. GLUT1. Its tissue location is brain, placenta, kidney, RBC. Its function is glucose uptake during fasting. GLUT2. It is located in the liver, pancreas, and the intestine. Its function is liver glycogen formation, pancreas insulin secretion. GLUT3. It is located in the brain, neuronal, placenta, kidneys. Its function is glucose uptake during fasting. GLUT4. It is located in skeletal muscles, cardiac muscles, and adipose tissues. Its function is insulin-stimulated glucose uptake after meals. GLUT5, located in the small intestine and in testis, in the sperms mainly. Its function is fructose transport. GLUT6 is located in white blood cells and the spleen. Its function is not known. GLUT7 is located in the liver endoplasmic reticulum, and its function is glucose transporter in the endoplasmic reticulum. Rate of glucose transport via GLUT depends on number of GLUTs, concentration of glucose, affinity of GLUT for glucose. Multiple choice question points. Widely distributed glucose transporter is GLUT1. Most abundant glucose transporter in red blood cells is GLUT1. Major glucose transporter of brain is GLUT1. Major glucose transporter of neurons is GLUT3. Major glucose transporter of placenta is GLUT1. Glucose transporter of blastocyst is GLUT8. Insulin-dependent glucose transporters are GLUT4, 8, and 12. GLUT1 is not present in the neurons. Urate transporter is GLUT9.